Types of white blood cells. Granulocyte. They are also called phagocytes or polymorphs. They have a large lobed nucleus and cytoplasm containing granules, hence the name granulocytes. They can change their shapes as they actively seek and engulf diseases causing germs in a process called phagocytosis, hence the name phagocytes. Some white blood cells may die in the course of phagocytosis. The dead phagocytes, together with dead microorganisms and damaged tissues, form pus. They can squeeze through capillaries walls in order to reach infected tissues. They are made in the bone marrow. Granulocytes. They have large rounded nuclei. Their cytoplasm is also non-granular. Types of granulocytes. Monocytes. They are formed in the bone marrow. They destroy microorganisms such as bacteria by engulfing them. Lymphocytes. They are formed in the lymph nodes and produce antibodies that protect the body from infections in the following ways. Antibodies which are antitoxic neutralize the toxins, antigens, produced by the pathogenic organisms. Some antibodies such as agglutinins cause clumping together of microorganism. This stops the microorganism from multiplying and eventually they die. They are then ingested by phagocytes. Lysins destroy microorganisms by digesting their cell membrane or walls. Opsonins are antibodies which adhere to the outer surfaces of microorganisms, thus making it easy for phagocytes to ingest them. Opsonins are only produced during infection. Mechanism of blood clotting and its importance. Platelets, thrombocytes. They are very small and have no nucleus. They are fragments of RBC and they are made in the bone marrow. There are approximately 250,000 platelets per cubic millimeter of blood. They live for about 7 days. Platelets produce an enzyme known as thromboplastin which plays a key role in blood clotting. Blood clotting. Blood clot is a seal that forms to close blood vessels that are cut or damaged. This has two functions. Stops further bleeding at the wound and therefore prevent excessive blood loss. Prevents entry of harmful bacteria into the body through the damaged tissue. Process of blood clotting. When the blood vessels are damaged, the damaged tissue and platelets release an enzyme called thromboplastin, thrombokinase. Thromboplastin initiates the process of blood clotting by neutralizing the anticoagulants factor known as heparin which occurs naturally in blood. Thromboplastin activates the conversion of prothrombin, blood protein, to thrombin in the presence of calcium ions. Vitamin K is required in the formation of prothrombin. Thrombin activates conversion of soluble fibrinogen which is an inactive protein to insoluble fibrin which forms a meshwork of fibers on the cut surface to trap RBC to form a clot. Blood groups, ABO system and the rhesus factor. Human blood can be grouped using the ABC system and rhesus factor. The ABO system. The ABC of humans has special types of protein called antigens. There are two types of antigens, i.e. antigen A and antigen B. Antigens determine the blood type or blood group of a person. A person with only antigen A on their RBC is said to belong to blood group A. People with antigen B. Sometimes both antigens A and B are found on the RBC of the individual. In such a case, a person is said to belong to blood group AB. In addition to the antigens on the RBC, blood plasma contains other types of proteins called antibodies. These are complementary to the antigens A and B. Antibodies are named A and B respectively. Antibodies and antigens do not correspond to each other, e.g. a person with antigen A will have antibody B in the plasma. A person with antigen B will have antibody A in the plasma. If both antigens are present as in blood type AB, then no antibodies will be present in the plasma. If none of the antigens is present, then both antibodies are present, e.g. in blood group O, e.g. blood group antigens antibody, AAB, BBA, ABA and B. None, O none A and B. NB. The presence of an antigen and its corresponding antibody in the blood of an individual would lead to clumping of RBC. This is referred to agglutination. Blood transfusion. It's the process of putting donated blood into a receipt. A blood donor is someone who voluntarily goes to a hospital or health center to give blood. The donor should be a healthy individual between 18 to 65 years. Blood is taken from the donor through a vein in the arm and passed into a bag containing anti-clotting substances. The blood is kept in bank under suitable conditions to be given to a patient who needs it, within I month, because RBC will have died after I month. A blood transfusion may be necessary in situations such as when a person loses too much blood due to an injury that may result from motor accident, or Kcse. When a person becomes anemic due to diseases such as malaria. When a woman loses too much blood after childbirth. When the blood of the donor and recipient mix freely without agglutination, the blood from the two individuals is said to be compatible. The blood from two individuals is said to be incompatible if agglutination occurs when the two blood are mixed. Individual with blood group AB can receive blood from all the four blood groups because AB has no antibodies to agglutinate the recipient's blood, hence referred to as universal recipient. Precautions before transfusion. The recipient must be given compatible blood, i.e. blood received by recipient should not agglutinate. Compatibility of blood is determined by A and B antigens and rhesus antigens. After somebody has donated blood, it's first screened before it is kept in a blood bank or transfused into a recipient. During screening, doctors test blood for presence of any infective microorganisms, e.g. HIV. If blood is infected, it's normally thrown away. After somebody has donated blood, she receives a blood donor card bearing the name of donor and her blood type. Rhesus factor. The RBC may also have another antigen on their membrane known as rhesus factor. Individuals with rhesus antigens on the membrane of RBC are said to be rhesus positive, Rh+, while individuals without the rhesus antigens are said to be rhesus negative, Rh. When a Rh woman marries a Rh+ man, the woman will conceive a Rh+ fetus. The Rh+ antigens of the fetus pass across the placenta into the mother's bloodstream during the last month of pregnancy. The mother responds by producing Rh antibodies which cross the placenta into the fetal circulation. The rhesus antibodies destroy some of the RBC of the fetus. The firstborn child has a higher chance of survival because the destruction of RBC is minimal. But in subsequent pregnancies, massive destruction of RBC occurs, leading to the death of fetus. This condition is referred to as erythroblastosis fetalis or hemolytic disease of the newborn. The mother can be treated with a rhesus globulin which prevents her from producing antibodies against the fetal antigens. This will protect the RBC of the fetus in subsequent pregnancies. Also, the baby is transfused with Rh blood after birth due to the extensive breakdown of RBC. Lymphatic system. Animals, particularly vertebrates, have an additional transport system besides the blood system. This is known as lymphatic system, and it supplies all the regions of the body just like the blood system. The lymphatic system is made of narrow, thin-walled tubes known as lymph vessels which branch to form lymph capillaries in which a fluid known as lymph is transported. Lymph. This is a fluid similar to blood plasma except that it contains less protein. It's formed as a result of ultrafiltration of blood from the narrow blood capillaries. As blood circulates, it reaches the body tissues through the blood capillaries that form a network throughout the tissues. The pumping force from the heart together with the narrow lumens of the capillaries exert a high pressure that forces the fluid part of the blood to filter out of the capillary walls into the surrounding tissues. This filtrate consists of all the constituents of blood plasma except the blood cells' proteins. This is because the blood cells and proteins are too large to filter out of the capillary walls. The fluid is known as tissue fluid or intercellular fluid. Once formed, the tissue fluid bathes the cells of the tissues, supplying them with O2, food and other useful substances. The cells absorb these substances and pass out CO2 and other waste products in exchange. Most of the tissue fluid then return to the blood system through the venule end of the blood at the capillary. The excess tissue fluid drains into the lymph vessels where it is known as lymph. Lymph vessels have a swelling along their length called lymph nodes. They contain lymphocytes which defend the body against infection by producing antibodies that kill bacteria. Also in the lymph nodes there are phagocytes that engulf bacteria. Immune responses. The microorganisms that cause diseases are called pathogens. The production of antibodies by special cells that inactivate foreign substances is called the immune response. The ability of the body to defend against infection by producing antibodies or cells that destroy pathogens is called immunity. The immune system includes all the parts of the body that are involved in the recognition and destruction of foreign substances. It's made up of bone marrow, which produces white blood cells. White blood cells, especially phagocytes and lymphocytes. Various tissues of the lymphatic system, such as lymph nodes, tonsils, thymus, and spleen, which accommodate lymphocytes. Types of immunity. They are classified into two major groups, i.e., innate, inborn, inherited. Acquired, natural and artificial immunity. Refers to natural and natural body defense like the skin, sebum and mucus, and sickle cell anemia. This type of immunity is dependent on genetic constitution of an individual, e.g. blacks are generally less susceptible to malaria than whites. Acquired immunity. A natural acquired immunity. This occurs when the body naturally overcomes an infection, e.g. Natural active immunity. This is the type of resistance which is built up in a person after suffering and then recovering from a disease. The person develops specific antibodies against future attack of these same pathogens. E.g. when a patient recovers from chicken pox, measles, he develops immunity against these diseases. A patient cannot suffer from reinfection. Natural passive immunity. It's the resistance which is inherited, i.e. passed on from parents to offspring via placenta or onto a newborn baby through colostrum. B. Artificial acquired immunity. This is the immunity acquired when the antibodies are artificially introduced into the body or weakened pathogens are introduced in the body. It's divided into artificial active immunity. It's developed by introducing a weakened dose of a microorganism into a healthy person to stimulate the immune system to produce antibodies and antitoxins. The process of weakening the disease-causing microorganism is known attenuation. The weakened microorganisms such as bacteria and viruses are given in the form of a vaccine. The immunity developed lasts for a certain period of time, e.g. immunity against cholera lasts 6 months while that for smallpox lasts several years. Artificial passive immunity. This is the immunity that comes from using antibodies produced in one organism to protect another organism from a specific disease, i.e. the immunity acquired when preformed antibodies are artificially introduced into the body of a patient. These antibodies are called antisera, e.g. anti-tetanus, antirabies and antivenom antisera. In this type of immunity, antibodies are administered to the body when it cannot form its own antibodies. This is common during a disease outbreak. The immunization is provided in the form of anti-serum. An anti-serum is a serum containing antibodies. It is administered in the case of tetanus, diphtheria, rabies and cholera. Immunity acquired this way lasts for a short time. Allergic reactions. At times the body's natural defense system may overreact against even harmless substances such as dust, pollen, certain food, insect stings and bites. Such substances are referred to as allergens, and they provoke the cells to produce and release chemicals such as histamine, which causes inflammation, itchiness and pain. Allergic reactions may cause skin rashes, itching, sneezing, vomiting, coughing and swelling of the body. A severe condition called anaphylaxis sometimes occurs, in which the blood vessels get dilated and this lower the blood pressure to the extent of causing death. This is how the bee stings can cause death. Doctor usually prescribe an antihistamine treatment to counteract the effect of histamine. Importance of vaccinations against diseases. Tuberculosis, poliomyelitis, measles, diphtheria, whooping cough. Protects individuals from infections, e.g. smallpox, tuberculosis, TB, E. T. C. Prevents the spread of diseases. A vaccine is a weakened or dead form of a disease-causing microorganisms. Vaccines are administered orally or by infection. The immunization program is carried out nationwide by the Kenya Expanded Program of Immunization, KEPI. Name of disease causative agent, age when administered. Method of vaccination. Tuberculosis, TB, bacterium at birth. Injection. Poliomyelitis, polio, virus at birth. 6, 10, 14 weeks. Oral inoculation. Diphtheria bacterium 6, 10, 14 weeks. Injection. Whooping cough bacterium 6 and 14 weeks injection. Measles virus 9 months injection. Practical activities. Observe permanent slides of sections of stems and roots. Carry out experiments to compare transpiration on lower and upper leaf surfaces. Observe wall charts, models. Analyze data on transpiration rate under different environmental conditions in plants. Dissect a small mammal and observe its transport system. Demonstration. Make a longitudinal section of the mammalian heart to display the chambers and associated blood vessels. Record pulse rate at the wrist before and after vigorous activities and analyze the results. Demonstrate the unidirectional flow of blood in the cutaneous veins of the forearm.